Yes, good evening everyone. Karanveer, Vidhi, Akash, Lakshita, Rafiq, Shailesh, Life and IMS, Arsh, Abhishek, yeah, Vipin, Fatima, sir is also here. Yeah, today we'll start the class. Yes, Sakib, Pratap, Kailash, very good evening, guys. Today we'll master the enzymes of replication. So just waiting for another 2 minutes so that everyone can join the class, and then we'll start with our today's lecture on DNA replication enzymes. Okay, we'll start with DNA replication enzymes today. That is the part 2. Okay. Yeah, Vijay, Rajput, Kanchandeep, Dipanshu, Gayatri, Shinam, Gaurav. Very good. Very good. Welcome, guys, to the class. Yeah. Ask everyone to join us. The class has already started now. Ask everyone to join as the class has already started. Nivedi, welcome. Yash, welcome. Welcome to the class, guys. So, yes. Yes, Arvind, Payal, Kanchandeep, Deva, very good, guys. Just waiting for another 2 minutes so that everyone can join the class, and then we'll start with the replication enzymes. Yes, yes. I'm very much good. Dev, Ajay, Abdul Talib, DJ, very good. Neo, Abhishek, very good, guys. So yes, Nidhi, very good, guys. You all have come like online to study with me, and today I like you will be great that we will learn the replication enzymes in a very um, easy manner. Okay, so yeah, so the very first thing that we will discuss today, it is about the different enzymes of DNA replication, and the very first one that you all should know, that is the topoisomerase. That is the topoisomerase, guys. And remember that topoisomerase, basically it prevents the torsional strain. It prevents the torsional strain. Okay, so whatever, like you already know that after unwinding of the DNA, basically when there is a formation of a replication fork, basically the two strands of the DNA has unwinded, the support has already gone. So ultimately to sustain that particular strain, basically this topoisomerases are there, basically so that the DNA strands can prevent the torsional strain. Okay, yeah, so ultimately, I hope the audio and visuals are clear to everyone. Guys, just give me a thumbs up that you all are participating. You are ready with your notes. You are ready with your pen, pencils, diagrams, everything. So yeah, just give me a thumbs up, guys. Yes. Okay. Very good. Nesi, very good, guys. I want every one of you to like be over here. Yes. Yes, Karanveer, very good. Okay. Coming on to the next enzyme, guys. Yes, Vidhi Rajput, very good. Coming on to the next enzyme, guys, that is the helicase. That is the helicase, guys. And remember that you already know what is the function of the helicase. It is the unwinding enzyme. It is the unwinding enzyme. I'm very happy that everyone is participating right now. Yeah, very good, guys. Yes. Yes, very good, guys. Good evening, guys. Please be with me throughout the lecture and you will learn. Helicase basically means the unwinding enzyme. Unwinding enzyme. You already know that the enzyme which actually unwinds the two strands of the DNA, that is the helicase. So, helicase is the unwinding enzyme. Coming on to the third enzyme, guys. The third enzyme, it is the DNA primer. Okay, DNA. Yeah, Okay. The third enzyme, it is the DNA primer. And yeah, okay. Firstly, we'll discuss about the, yeah, DNA ligase. You already know that DNA ligase is basically responsible for, already know that it helps in the sealing of the nicks. It helps in the sealing of the nicks. Yes, Anshay, welcome to the class. Kindly take out your notes, Lakshay. Yeah, take out your copy books and start writing the notes, guys. Very, very important topic today I'm discussing. That is the enzymes of DNA replication. And yeah, so you should know that the DNA ligase is basically helpful in sealing the nicks. You already know that whatever gaps, suppose this is the replication fork and this is the lacking strand synthesis, okay? You already know that whatever gaps are present, okay, they are being added by the DNA nucleotides, okay, and ultimately they are joined together by DNA ligase. So DNA ligase is like a fevicol, okay. Basically it helps in sticking the all the pieces of the newly synthesized DNA, basically the Okazaki fragments together, okay. That's why it is the fevicol of the molecular biology, we say. So DNA ligase is for that only, okay. Apart from that, guys, remember that we have an enzyme that is called as DNA polymerase, that I will take it separately right now. And you already know that DNA polymerase cannot act like roughly, it cannot act somewhere. So it needs an RNA primer to show its particular action. So ultimately, DNA polymerase is involved in the synthesis of the DNA and synthesis of the new strands. Synthesis of the new strands. So yeah, synthesis of the DNA. So ultimately, yeah, it is there. Apart from that, guys, remember that, yeah, now we will talk about DNA polymerase in detail. We'll talk about DNA polymerase in detail, guys. So put the heading as DNA polymerase. I hope everyone is participating, guys. Everyone is participating. Just give me a thumbs up, guys. You all are with me. You are writing. You are like watching the video. And yeah, everything is clear as of now. Chalo, very good, very good. Guys, like I really want everyone to join the class. Yeah. Yes, very good. Very good. Very good, guys. Very good. ओके, चलो बच्चे, हेडिंग डालो डीएनए पॉलीमरेज, ओके। तो डीएनए पॉलीमरेज बच्चे हम लोग यूकैरियोट्स में पढ़ेंगे एंड वी विल सी द डीएनए पॉलीमरेज, डिफरेंट टाइप्स ऑफ डीएनए पॉलीमरेज इन प्रोकैरियोट्स आल्सो. So firstly, you all know that we have the prokaryotes, okay, which do not have a very well defined nucleus. The nucleus is nearly scattered all around the cytoplasm. That is the prokaryotes, and that is like the primitive organisms basically show this type of nuclear behavior, and it's almost typical in bacteria and all. And coming on to the eukaryotes, the higher cellular organisms, you means we are having a true nucleus which is bounded by a nuclear membrane, which is bounded by a nuclear membrane, guys. Yeah, which is bounded by a nuclear membrane. Which is bounded by a nuclear membrane. So ultimately, eukaryotes and prokaryotes, basically we have different types of DNA over here. We have different types of DNA. So remember, in prokaryotes we have three types of DNA. So kindly mention the names and then we'll discuss the functions as well. So we have the DNA polymerase 1, DNA polymerase 1, okay. Then we have DNA polymerase 2, DNA polymerase 2, and we have DNA polymerase 3, DNA polymerase 3. Coming on to the eukaryotes, guys, we have 5 types of different kinds of DNA polymerase that you all should remember. And yeah, the 5 types basically are the DNA polymerase alpha, DNA polymerase alpha, DNA polymerase beta, DNA polymerase gamma, DNA polymerase delta, and we have DNA polymerase epsilon. So basically we have 5 types of eukaryotic DNA polymerase starting from alpha, beta, gamma, delta and epsilon. So we will discuss all these in detail, guys. We will discuss all these in detail. So kindly make this particular table that DNA polymerase is categorized like in prokaryotes it's of 3 types, in eukaryotes it's of 5 types. It's of 5 types. It's of 5 types. Yes. Guys, be in the class and just try to master the DNA polymerase. First we will discuss about the different types of DNA polymerase in prokaryotes. So now guys, put the heading as DNA polymerase 1. Yeah, put the heading as, yeah, Ronak, Sagar, Ankit, Sachin, Ritik, welcome to the class. Kindly make the notes along with me. It's not for your attendance or something. You have to be in the class, write the notes and send my notes, send all the notes whatever you have written during the live lecture to my WhatsApp. And instant sending the notes is like like a proof that you have attended the lecture live. So starting with the DNA polymerase 1. Starting with the DNA polymerase 1, guys. Remember, now what is the function of the DNA polymerase 1? Now DNA polymerase 1, basically it is involving in the removal of primers. Removal of primers, guys. Removal of primers. You guys already know that we have the replication fork, and for the new strand to be synthesized, we need the RNA primers. We need RNA primers. You already know that DNA polymerase 3 cannot act on this or any rough scratch. So basically a RNA primer is required. So since RNA primer is there, and after that you already know that the DNA polymerase 3 will act and we will have the new strand synthesized by DNA polymerase 3. So ultimately this RNA primer is not required in this new DNA. ये RNA primer की तो जरूरत है ही नहीं DNA में. So so the removal of these primers is basically done by DNA polymerase 1. This removal of these primers are done by DNA polymerase 1. So that is the first point that you have to mention in your notes. Coming on to the second most important function, DNA polymerase 1 basically also helps in gap filling. It also helps in gap filling. You already know that on the lagging strand, we have the Okazaki fragments, and in between we have gaps. So ultimately, these gaps are basically filled by DNA polymerase 1. These gaps are filled by DNA polymerase 1. Yes. So apart from that, guys, remember that it is involved in other 2 functions as well. It is the DNA proofreading function, DNA proofreading, and the last, it is the DNA repair. Repair of DNA I will take in another lecture and write down major. DNA repair, majority of the DNA repair, it is done by DNA polymerase 1. It is done by DNA polymerase 1. Okay. Before launching the DNA, basically the DNA is checked whether it is functional, non-functional, its base pairs are like accordingly arranged. So ultimately the proofreading of the DNA, it is also done by DNA polymerase 1. It is already done by DNA polymerase 1. Yes. Coming on to the next, that is the DNA polymerase 2, guys. Coming on to the next, that is the DNA polymerase 2. And you already know that DNA polymerase 2 is also involved in DNA proofreading, and it is also involved in the DNA repair. It is also involved in the DNA repair, and that is the minor function majority of the DNA repair it is done by DNA polymerase 1, but minor DNA repair it is done, like some of the DNA repair it is done by DNA polymerase 2. Now coming on to the third one, that is the DNA polymerase 3. Third, that is the DNA polymerase 3. DNA polymerase 3, guys. And you already know that it is involved in the leading strand synthesis. It is involved in the leading strand synthesis. Apart from that, you should remember, I already told you that suppose this is the replication fork and this is the RNA primer. ठीक है, तो ultimately the leading strand will be formed by the action of the DNA polymerase 3. Okay, and that's to the function of the DNA polymerase 3. It helps in the leading strand synthesis. Okay, apart from that, it also helps in Okazaki fragment synthesis. I have already told this you in my previous video. Okazaki fragment synthesis, and it is also involved in DNA proofreading. It is also involved in DNA proofreading. It is also involved in DNA proofreading. Now some of the questions which have been asked in the NEET PG exam, guys, and that I wanted to mention with a different color so that you can all mark this with a dollar. Okay, so remember that it is the most processive or most processing or processive DNA polymerase. तो सर इसका मतलब क्या होता है? You already know that कि once DNA polymerase binds to the DNA, it will add maximum number of nucleotides. Okay, so ultimately you सबसे ज़्यादा प्रोसेस करवाता है क्योंकि ये लीडिंग स्ट्रैंड को भी सिंथेसाइज करता है एंड अल्टीमेटली इट सिंथेसाइजेस द ओकासाकी फ्रैगमेंट्स आल्सो. So it is the most processive DNA polymerase. It is actually helping in the leading strand synthesis and the Okazaki fragment synthesis. DNA polymerase जब भी DNA को bind करेगा, it will add maximum number of nucleotides. और maximum number of nucleotides मतलब it is the most processing part. Okay, so that's why it is known as the most processive DNA polymerase. Apart from that, guys, just give me a thumbs up that you all are over here. You're writing the notes and would be sending the notes to my WhatsApp after the class, suppose. Yeah. Ignore the unethical students जो ये mad machine है. नाम तो अच्छा लेकर आया करो ना लाइव चैट क्लास में, आते ही वो डिस्टर्ब करने, तो नाम तो कैची रखा करो ना. हमें पता है तुम पागल हो और पागल को चैट बॉक्स में अपने आप को पागल करने साबित करने की ज़रूरत नहीं है. सो मतलब हो तो तुम स्टूडेंट ही कोई. मतलब नए नए पैतरे जो रहते हैं ना, बिल्कुल कौन सा सीरियल देख के ये सब दिमाग लगता है तुम्हारा. लेट मी नो द नेम ऑन माई व्हाट्सएप. एनीवेज चलो कुछ नमूने आते हैं बट विल ट्राई टू इग्नोर दैट. या now guys, I wanted to highlight some important points on DNA polymerase 1. Now this DNA polymerase 1, just try to mention some point that DNA polymerase 1 remember it is DNA polymerase 1, it is also called, okay, it is also called as Kornberg's enzyme. Ashok, good evening. You are late. Try to write the notes along with me. Okay, it is also known as Kornberg's enzyme. It is also known as Kornberg's enzyme. Why it is known as Kornberg's enzyme? Because this was discovered by, Kornberg, or we can simply say the Arthur Kornberg, okay, in E. coli bacteria. Apart from that, guys, yeah, it is also known as clinopolymerase. It is also known as clinopolymerase. It is also known as clinopolymerase, and you already know that basically it helps in, or yeah, so it helps in basically the 5 prime to 3 prime exonuclease activity is removed. Okay, yeah. Yeah, it's just on the scientist name. Yeah. Exonuclease activity removed, in removal of this exonuclease activity, basically DNA polymerase 1 is also functional. We'll just learn it in another few seconds, a few minutes. Okay, just try to mention the points as of now. And now guys, the very important part that I wanted to mention, yeah, it is the eukaryotic DNA polymerase, and in one slide we will learn the complete eukaryotic DNA polymerase, and just try to draw this eukaryotic DNA polymerase with me guys. Eukaryotic DNA polymerase. Eukaryotic DNA polymerase. Now guys, how to learn this कि सर ये तो बहुत टफ होगा, इतना 5 पार्ट्स हैं, I just don't know how to learn this, so I'll just try to simplify this guys for you people. Guys, we will have 2 sets over here, the starting point, and we will have the finish point. I don't know why. Yeah, we will have the finish point guys. We will have the finish point. We have 2 points, and just I will try to tell you how all the functions of the eukaryotic DNA polymerase in just this simple diagram. You already know that. Okay, now suppose let's just take the example of a race. Okay, in the race we have the starting point and we have the finish point. Okay, a simple race, that is a sports. Okay, the simple race, we have the starting and the finish point, and we will now try to arrange all the eukaryotic DNA polymerase, that is the alpha, beta, gamma, delta and epsilon, just in the series that I have drawn over here. And now guys, try to learn along with me. Can you see that which now guys, my question is, which DNA polymerase is basically leading, or it's first in this particular diagram? Now my question is, which particular DNA polymerase out of these 5 it is leading, or it's like number 1 in the race? So let me have the answers in the chat box, and then the function is quite easy guys, Yeah, waiting for the answers. Yeah. Just see, visualize and try to answer guys. Yeah, waiting for the answers. I perfectly want each one of you to answer the question. नहीं बच्चा, विधि, अल्फा, राजपूत, राहुल, तुम लोग क्लास में ही नहीं थे ना, मतलब फिनिश पॉइंट के पास एप्सिलॉन है कि अल्फा है? हु इज लीडिंग द रेस यार? तुम लोग कैसे आंसर्स बोल रहे हो? Lost. DJ, very good. Yeah, the last one, that is the epsilon guys, and it is leading all from all the 5. Yeah, it is leading from all the 5. Yeah, it's the epsilon guys. And remember the epsilon, write down the function guys, very easy. It involves in the leading strand synthesis. Yes, it involves in the leading strand synthesis guys. Epsilon, very good. The 5th one guys. Now guys, which one is lagging just before the leading strand? My question to you next, which one is lagging just before the leading strand? I'm waiting for the answers guys. Which one is lagging just before the leading strand? Yes. Just waiting for the answers, guys. I cannot see the comments. Hmm. So the one which is just lacking behind the leading one, it is the delta. So delta, DNA polymerase delta, yes, Kanchandeep, very good, Pacha. Karanveer, very good. So delta, DNA polymerase delta, basically it helps in the lagging strand synthesis. It helps in the lagging strand synthesis, guys. वेरी इजी कि भाई एक हमने रेस का वो बना लिया है. वी हैव द 2 points, द स्टार्टिंग पॉइंट ऑफ द रेस, दैट इज द स्टार्टिंग पॉइंट. यस, एवरीवन इज राइट. डीजे प्रसन्ना, विधि, अमजद, अब्दुल, परमजीत, रौनक, मनीषा, इंशा, सोनू, उमा, वेरी गुड गाइस. Very good guys. Like I'm truly happy that at least whosoever you are like participating in my class, I assure you that you will clear the exam in one go. If you are constant throughout the lectures, if you are making the notes along with me, you are like perfectly learning along with me right now. Okay, so yeah, it is the lacking strand synthesis, just lagging behind the epsilon. So yeah, apart from that guys, remember that gamma is basically situated in the middle. Gamma, it is basically situated in the middle. So it is in the middle position. You can see that 2 are above, 2 are down. Gamma, it is in the middle position. So DNA polymerase gamma is basically for mitochondrial DNA synthesis. It is in the middle position ना, middle में है M और ये वाला M. So middle basically mitochondrial DNA synthesis guys. Mitochondrial DNA synthesis. This question has been asked multiple number of times, that which DNA polymerase is actually helping in synthesis of the mitochondria. Okay, so that's again very easy for you guys. That's again very easy for you guys. Yeah. Yeah. So ultimately after that guys, just try to see that what is the first step. You already know that कि अल्फा जो है सर वो तो स्टार्टिंग पर ही है अभी. तो you already know that what is the first step of the DNA synthesis. Okay, what is the first step of DNA replication guys? Synthesis of DNA के लिए what is the first step guys? Apart from, yeah, so ultimately it helps in synthesis of RNA primer. RNA primer ऐड होगा, उसी के बाद तो DNA polymerase 3 एक्ट करता है to continue the leading strand, and that's why it is on the first position. And we have the first step guys, that is the synthesis of the RNA primer, that is the alpha, that is synthesis of the RNA primer, that is synthesis of the RNA primer guys. Apart from that guys, yeah, the beta one that you guys have to remember. Okay, beta is basically the major DNA repair enzyme. It is the major DNA repair enzyme. So just comparing it the fact कि beta और बेटी, you already know that कि beta or बेटी, it basically depends on the DNA of the parents. So ultimately the majority of the DNA, basically beta और बेटी का सारा majority of the DNA depends on the parents. So beta is basically the major DNA repair enzyme. That is again a question. All 5 questions, all 5 functions done in one single slide. Now I will add some more function guys. I will add some more function. I hope everything is clear to everyone. Everything. निवेदी, तुम लोग को पता ही नहीं चल रहा है कि डेल्टा वाला कंसेप्ट तो कब का चला गया, अब हम लोग कितना आगे बढ़ चुके हैं. पता नहीं कौन से तुम लोग. एनीवेज, yes, Now guys, remember that, yeah, the last three one basically it is involved in the proof, reading, synthesis mechanism, proof, reading, synthesis mechanism, and yes. So ultimately लास्ट के तीनों देखो सिंथेसिस के काम आ रहे हैं. माइटोकॉन्ड्रियल डीएनए भी सिंथेसाइज हो रहा है, लैकिंग स्ट्रैंड की भी सिंथेसिस हो रही है और लीडिंग स्ट्रैंड की भी सिंथेसिस हो रही है. सो अल्टीमेटली प्रूफ रीडिंग, ओके, और जो भी सिंथेसिस मैकेनिज्म है सारा का सारा लास्ट के तीन वाले देखेंगे, सारा का सारा लास्ट के तीन वाले देखेंगे. So ultimately, yeah, everyone, yeah, like I'm quite happy that every one of you are like, yeah, yeah, proofreading or the synthesis mechanism. So that is all the eukaryotic DNA polymerase जितना आप लोगों को पेपर के लिए याद रखना है. बहुत ही सिंपल तरीके से एक सिंपल डायग्राम में सारे फंक्शंस कवर हो गए और ये हमेशा नीट के एग्जाम में, पीजी के एग्जाम में, एफएमजी के एग्जाम में बहुतों बार पूछा गया है. सो मार्क द स्लाइड आज, लाइक इस पे $1,000 लगा लो क्योंकि दिस इज वेरी वेरी इंपॉर्टेंट. दिस इज वेरी वेरी इंपॉर्टेंट गाइस, दैट इज वेरी वेरी इंपॉर्टेंट. या गाइस, coming on to the differences between the eukaryotes and prokaryotes, the replication guys. Now remember guys, the very first point, that is the origin of replication. Okay, I already told you in prokaryotic cells we have one origin of replication, okay, and in prokaryotic and the eukaryotic we have multiple origin of replication sites. We have multiple ORI in eukaryotic and a single ORI site in the prokaryotic site. Unwinding DNA double helix structure, it is done by helicase in prokaryotic cells. It is done by helicase in eukaryotic cell as well. Okay. So remember the stabilization of the unbounded template strands. I already told you कि single stranded binary proteins or binding proteins are there. Okay. And it is the single stranded binding proteins in eukaryotic cells also called as the replication protein A. Also called as the replication protein A in case of eukaryotic cells. In case of eukaryotic cells. Apart from that guys, I have already told you कि suppose अगर ये दो strands हैं और ये दो strands आपस में they have got unwinded, अब ये दोनों वापस में आकर मिले ना, इसका ख्याल कौन रखता है? that is the single stranded binary protein. अब इसको तुम, अब suppose ये दिस इज अ बॉय, दिस इज अ गर्ल, बॉयफ्रेंड और गर्लफ्रेंड है, ठीक है, और सिंगल स्टैंडर्ड बाइनरी प्रोटीन होता है गर्लफ्रेंड का पक्का वाला दोस्त. तो अब ये क्या करता है कि एक बार जब गर्लफ्रेंड और बॉयफ्रेंड में ब्रेकअप हो गया तो ये जो सिंगल स्टैंडर्ड बाइनरी प्रोटीन है ये गर्लफ्रेंड को वापस बॉयफ्रेंड से मिलने नहीं देगा, वो टेक ओवर कर लेता है गर्लफ्रेंड को. सो अल्टीमेटली सिंगल स्टैंडर्ड बाइनरी प्रोटीन ऐसा स्ट्रक्चर है that will not allow these two strands to meet together. Ultimately उनका जो ब्रेकअप हो गया है वो मेंटेन्ड रहे, उसका पूरा ख्याल रखता है उसका पक्का वाला दोस्त. So that is the, yeah. Apart from this, remember the synthesis of the RNA primer, it is the primase activity. Okay, the synthesis of the DNA, you already know that DNA polymerase 3, and over there also we have the alpha plus delta part over there. Okay, the alpha plus delta part, majority it is the DNA primers which are synthesized. Okay, and you can just mention it over here. DNA alpha, basically it is the synthesis of the RNA primer, it is synthesis of RNA primer. Okay, and delta 1, basically you already know that it is in the lacking strand synthesis, it is in the lacking strand synthesis. Plus epsilon, we have the leading strand synthesis, and the gamma 1, you already know that it is the mitochondrial synthesis. It is the mitochondrial synthesis. Yes, it is the mitochondrial synthesis guys. Apart from that, removal of RNA primers, it is by DNA polymerase 1, okay, that acts in the 5 prime to 3 prime exonuclease. I just mentioned in like a previous slide also. And over it is the RNase H that is also acting in 5 prime to 3 prime exonuclease. That is also acting in, yeah. So yeah, रिप्लेसमेंट ऑफ आरएनए विद डीएनए, मैंने बोला था आरएनए प्राइमर को लाइक कट कर कर निकाल देंगे और उसकी जगह डीएनए ऐड करेंगे. दैट इज बाय द डीएनए पॉलीमरेज 1 ओवर हियर एंड डीएनए पॉलीमरेज डेल्टा इन यूकैरियोटिक सेल्स इन लैगिंग स्ट्रैंड सिंथेसिस. आई हैव ऑलरेडी टोल्ड यू लैगिंग स्ट्रैंड में ओकाज़ाकी फ्रैगमेंट्स बनते हैं और बीच में जो स्पेसेस होते हैं आरएनए प्राइमर निकालने के बाद उन स्पेसेस को फिल करने का काम भी डीएनए पॉलीमरेज डेल्टा का है इन यूकैरियोटिक सेल. दैट्स व्हाई आई मेंशन्ड इट इज इन्वॉल्वड इन द लैगिंग स्ट्रैंड सिंथेसिस गाइस. And joining of the Okazaki fragments, I already told you Fevicol of the molecular biology, okay, that is the DNA ligase. It will help the like pieces or the base pairs, basically everything to get like stuck together, okay. All the Okazaki fragments will be like they will stick together and they will form a continuous strand later on, okay, that will form a continuous strand later on. So that is the differences between the prokaryotic cell and the eukaryotic cell guys, okay. I hope this is clear to each one of you. All the enzymes which I have talked about right now, DNA polymerase, the different types and the different enzymes of DNA replication, everything is clear as of now. Just give me a thumbs up guys that you all are with me and all have written the notes along with me guys. Yes. Sure. Sure. Very good. Guys, now I will try that you should actually solve these questions. Question number one. Question number one. Yeah, I'm truly happy that all of you understood the part today. Yes. Okay. Okay, guys. Very good. Very good. Very good, guys. Now guys, the very first question, it says that an increased melting temperature of duplex DNA results from a high content of adenine plus guanine, thymine plus cytosine, cytosine plus guanine. Mohammad Amjad says C. Okay, what about rest guys? What about rest? Okay, what about rest guys? सचिन 80 रिच एरिया नहीं होगा बच्चे. 80 रिच एरिया तो वो तुम्हारा आंसर तुम्हारे हिसाब से तो वो ऑप्शन में ही नहीं है. तुम्हारे हिसाब से तो तुम्हारे लिए तो अलग से ना एक ऑनलाइन टेस्ट में अलग से एक वो ऑप्शन मार्क बनवाएंगे. वो ऑप्शन में ही नहीं है. चलो, व्हाट अबाउट रेस्ट गाइस? बाकी है भी कोई क्लास में आंसर देने के लिए? Karanveer says C. Okay, let's see why not other options. Vani Insha also says C. Sheenam says C. Okay, Sheenam says C. Abdul Lakshita also says C. Now Abbas B. Thymine cytosine. मनीषा वेरी गुड. सी बच्चे आप लोग एक बात बताइए, जस्ट टेल मी अ सिंपल थिंग कि सपोज दिस इज एडेनिन एंड थाइमिन, वी हैव टू हाइड्रोजन बॉन्ड्स and we have guanine and cytosine, we have 3 hydrogen bonds. कौन से वाले को तोड़ना, कौन से वाले को ब्रेक करना सबसे इजी है? Which one is basically easier to break? The one with 2 hydrogen bonds. Now the question says increased melting temperature. हमें टेम्परेचर किसके लिए ज्यादा चाहिए होगा? AT को ब्रेक करने के लिए या GC को ब्रेक करने के लिए? Yes, GC को ब्रेक करने के लिए टेम्परेचर ज्यादा चाहिए होगा ना, क्योंकि यहाँ पे 3 हाइड्रोजन बॉन्ड्स हैं. यहाँ पे 3 हाइड्रोजन बॉन्ड्स हैं. So ultimately high content of GC basically requires increased melting temperature, or I can say high melting temperature. So ultimately the answer will be C, guys. Ultimately the answer will be C. Yes, cytosine and guanine, guys. Answer is C. It is cytosine and guanine, guys. Yes. Obviously, I already told you that, yeah, everyone is right now. Yeah. So that is the concept, guys. That is the concept. जितना ज़्यादा bond, उतना ज़्यादा temperature चाहिए, उतना ज़्यादा heat चाहिए उसको तोड़ने के लिए. And it, yeah, very good. Very good, guys. Coming on to the next question. Coming on to question number 2, guys. It says that in RNA molecule, guanine content does not necessarily equal to its cytosine content, nor does its adenine content necessarily equal to its uracil content. Why? Or since it is a single strand molecule, double stranded molecule, double stranded helical molecule, or polymer of purine and pyrimidine ribonucleotides. Waiting for the answers, guys. Read the question and just let me know the answer, guys. Yeah. Yeah, Abdul Talib, very good. Yeah, it's more strong bond. Yes. Already discussed. Coming on to this particular question. Waiting for your answers, guys. क्वेश्चन कह रहा है आरएनए मॉलिक्यूल है, गुआनिन जो है वो साइटोसिन के बराबर ना, यू ऑलरेडी नो दैट जी बॉन्ड्स विद सी एंड एडेनिन बेसिकली बॉन्ड्स विद यूरासिल विद 2 हाइड्रोजन बॉन्ड्स, वी हैव 3 हाइड्रोजन बॉन्ड्स, यूरासिल इज प्रेजेंट इन आरएनए. सो अल्टीमेटली इट्स ट्राइंग टू आस्क कि why it is not necessary equal, ऐसा क्या रीजन हो सकता है. That's the simplest answer. It must be a single stranded molecule. We have the single stranded RNA. सिंगल स्ट्रैंड में एक ही स्टैंडर्ड रहेगा, तो अल्टीमेटली द कंटेंट्स आर नेसेसरीली नॉट इक्वल. ओके, इट्स अ सिंगल स्टैंडर्ड मॉलिक्यूल. यस, वेरी गुड. करणवीर, अर्श, सचिन, अमजद, लक्षिता, वेरी गुड. Rahul, Fatima, Prasuna, Shinam, Nimi, very good. Very good, guys. I'm truly happy that you all are present and attending the class live. Yes. I'm telling you, discussing questions after the class, it's very essential, guys. It's very essential. Coming on to the third question, that is the after formation of replication fork. Now the options are both the new strands are synthesized discontinuously, one strand is synthesized continuously and the other discontinuously, both the new strands are synthesized continuously, RNA primer is required only for the synthesis of one new strand. So the answers, guys. Yeah, just waiting for the answers, guys. Yeah, Karanveer says B. Okay, Sachin, chalo Sachin, you are now in the answer range. Fatima B, Abdul B, Neha B, very good, guys. Arsh B, very good. Heather, Prasuna, okay, Abdul, Amjad, Neo, Soumya, everyone says B. Very good, guys. I mean, you all have attended the lecture, so it's like a piece of cake for you guys. You already know that. Now, coming on to the replication fork, one is synthesized continuously, that is the leading strand, and one is synthesized discontinuously, that is the lagging strand. And synthesis always happens in 5' to 3' direction, and ultimately it is the semi-discontinuous nature of the replication. And I have already discussed semi-discontinuous का मतलब है कि एक तो सर पूरा सिंथेसाइज हो रहा है, कंटिन्यूस सिंथेसिस है, और एक डिसकंटिन्यूस सिंथेसिस है. सो सेमी डिसकंटिन्यूस भी बोल सकते हैं और सेमी कंटिन्यूस भी बोल सकते हैं उसको. ओके, वाइस वर्सा. So the very first option, it says that both the new strands are synthesized discontinuously. Wrong. You already know that one is synthesizing continuously. Coming on to the third, both the new strands are synthesized continuously. No, the lagging strand is synthesized discontinuously. RNA primer is required only for the synthesis of one new strand. No, leading strand में भी RNA primer की जरूरत है. Single RNA primer is required over here. Single RNA primer. और अल्टीमेटली लैगिंग स्ट्रैंड में मल्टीपल आरएनए प्राइमर्स की रिक्वायरमेंट है. मल्टीपल आरएनए प्राइमर्स आर रिक्वायर्ड. multiple RNA primers are required. You already know that we have multiple RNA primers. So RNA primer की जरूरत तो दोनों strand में, it's not only one strand. So this statement is also false. This statement is also false. So yeah, RNA primer is required in both the strands. Leading strand में single RNA primer is required, whereas when we compare it with the lagging strand, multiple RNA primers are required for the synthesis of the lagging strand. Okay. So yes, everyone is right over here. Everyone is right. Very good. Very good, guys. Very good. So yeah, everyone is right over here. I hope everyone understood the today's lecture very well. We have discussed 3 questions along with the differences in the replication of prokaryotes and eukaryotes. Okay, the steps or replication, the enzymes. Okay, ultimately the eukaryotic DNA polymerase. In one single slide I have just covered all the functions with very good tricks to remember. Okay, apart from that, all the functions are being discussed, guys. All the polymerases, all the enzymes are discussed. So yeah, I hope everyone understood today's lecture very well. So yeah, thank you for everyone like to participating, for your patience, for listening me out, guys. So like if you just meet me daily for like 40-50 minutes also, in like during whole day, you can actually compensate 40-50 minutes. So we'll take small topics and we'll try to cover the course of molecular biology, the anatomy daily. It's a free lecture, guys. So I truly appreciate that you should take the advantage of these live classes, because for you people, like, to understand things in a very conceptual manner, it is required. It is required. So yeah, thank you everyone for participating today. And tomorrow I will like take replication of DNA, or like we'll have, or no, we'll actually take the repair of DNA tomorrow, or telomerase. We'll just try to wind up the replication part and we'll see. Yeah. Okay. Yes, today's lecture is just a small lecture to complete the replication part. Yes. Okay, guys. What about my gift you have said? Sachin, इसका जवाब मैं तुम्हें WhatsApp पर दूंगा. तुम्हें जो चाहिए वो तुम्हें मिल जाएगा ज़रूर. Anyways, Shinam, very good बच्चे. Thank you guys. I like सर आपने डॉग स्मयन की आईडी ही क्यों बनाई है? Smayan is my nickname. That's why. Amjad. Abhijeet तो यार CID वाला भी फेमस है ना, तो ultimately I want myself to be unique. So yeah, it's Smayan means smile. इसका मतलब होता है मुस्कुराहट, खुशी. ओके, चलो वानिंशा, मज़ा आ गया यार, मज़े के लिए हूँ, मज़े में पढ़ लो. इट्स लाइक ऑल गुड फॉर यू गाइस. So thank you for participating, guys. Yeah, चलो ज़्यादा बातें नहीं करेंगे. मेरा चैनल like so waiting for the notes. You will, like, you promise me to send me the notes on my WhatsApp right now. Just waiting for it, guys. Waiting for it. Send me the notes. Yeah. Okay. Okay, guys. Goodbye. Amir Hassan. Okay, Amir, I don't know you. Text me on WhatsApp with your notes. Okay? Chalo. Good. Chalo. Bye.